And welcome back to Jeff Koinange live here at Citizen Television. What an interesting conversation we're having. It's about GMOs. What do you know about GMOs? Well, Professor Richard Oduor is here to talk about it. Dr. Roy Mugira, Kipruto Kiro, former agriculture minister, and miner, also here talking about GMOs. And joining us on Zoom is Dr. Bernard Muia. He's a public health expert. He used to be with the Ministry of Health. Dr. Muia, let me ask you real quick. When you heard that the ban had been lifted on GMOs, what were your thoughts?
Thank you so much for having me, Jeff. It's unfortunate that I was not able to join you, but I could hear you. I was following the conversation all the way through. I have several issues with removing the restriction about importation, cultivation of GMOs. And my basic issue here is what scientific evidence do we have that we don't have what has been stated by several of us in this conversation? Because my big problem is, what is the impact of GMOs in human beings? What evidence research have we subjected ourselves to prove that GMOs cannot cause serious public and medical issues? I have heard several people talking about some years that research has been conducted about the importance of GMOs. Personally, I seriously take issues of biotechnology as a medical doctor seriously. And there is serious room for us to use it for food safety and, of course, improving the nutritional status of our communities.
Okay.
But the basic question, Jeff, have we done any clinical trials? Which trials have we done for us to show that genetically modified foods are safe, and not a short-term research? We need a prospective studies, and those studies should be covering throughout the world globally, not in Kenya. And I will use a very simple word. We think globally, act locally. Where in the world have we subjected GMOs to those trials to prove that they cannot affect health of individuals and communities?
All right, Dr. Muia, I'll go straight to Professor Richard Oduor. You can answer that question.
I can.
Go on. Clinical trials or impact on human beings?
Jeff, I take particular concerns with Dr. Muia's actually communication here. Why? Because they have been, the medical doctors, have been prescribing genetically modified insulin even when there was ban. And he's not talking about it. And particularly, Jeff, let me probably expose a little bit about my my expertise in this medical field. My postdoc was with Pfizer in Sandwich, UK. And it was, Pfizer is one of the pharmaceutical companies. And we were making drugs for malaria, sleeping sickness, and research on cancer. So I have a fair understanding of how these things goes from preclinical to all the phases of clinical trials up to when it is out there. What they don't know is that even GMO, once it is released by this gentleman here, out there is post-release monitoring that also happens just the same way it happens in pharmaceutical world. It does happen. But let me tell you, Jeff, and this is almost hypocritical, because initially, any person who is alive right now and is suffering from diabetes, Jeff, world over, there is no any other source of insulin. All of it is genetically modified from a bacteria, E. coli. And how did it happen? Just a little bit of history so that people understand that it's really convenient for a doctor to talk very firmly on GMO while at the same time prescribing and GMO insulin and without blinking the eye. This is the thing. Before, when people were diabetic, we used to extract actually insulin from pigs. And what happened at that time? Do you know how many pigs we would have killed now if that technology, if GMO technology wasn't adopted. And what would it happen? We will get a chance. What would it happen if to the Muslims? Because they won't touch that insulin, however good it is. Now, this is what we did in genetic engineering. We said, Jeff, you are not diabetic. It means your insulin is correct. What is the gene in you that actually expresses and leads to the production of this right insulin? That is the gene that was specifically picked and cloned into an E. coli. E. coli is already part of the normal flora that we have in our stomach. So it's nothing new. Then you clone that in what we call in the lab expression cassette, transfer it into this E. coli, and tag it so that it excretes it as waste. It doesn't actually know that it's producing insulin for us. Then that is what, through industrial biotechnology, we purify and then now prescribe. That is GMO, recombinant DNA technology, swiftly. It is the same thing we used recently during the corona. That was RNase technology. It is still and they were here, and they actually supported the whole world.
Between red GMO, which is medicine, and apart from the COVID situation where the vaccines were produced too fast, in normal medical facilities or research, before a drug is released, it takes about 8 years. For GMOs, that does not happen. So I've talked about red GMO, but there's now green GMO is the agricultural part, which is very controversial, and we are against. For health, that is allowed because it takes longer research, 8 years. In fact, there was, you see, like when they do a lab test on rats, the lifespan of a rat is 2 years. So there are no clinical trials.
I want to
Dr.
touch,
Roy,
I want to touch something. Yes,
go.
Not going to that of the insulin, but I'm a regulator, and therefore I must be seen to be regulating the work that is coming from the labs and that which is coming out, and also addressing the concerns that are coming from our colleagues on this other side. Having said that, it is also my duty to point out that which is not correct, or that which is causing a little more confusion. The issue that Anne pointed about the Burkina Faso story, the short length of cotton lint, really had nothing to do with the GM technology, because the technology that was used for that cotton is the BT cotton to resist the cotton bollworm. So it was a breeding that had gone wrong. It had nothing to do with the GMO. She spoke about the cost of seeds, which again, Mheshimiwa has pointed to, which I would want to touch on a little. Now, when the Burkina Faso people are growing BT cotton, they would reduce the number of sprays for pesticides, which again, Anne is calling chemicals, and there is no category of all chemicals, that GMOs are not produced using chemicals. In fact, the BT cotton will reduce the number of sprays from 12 to only about 3, targeting other pests. And when you pick a sprayer carrying, for example, an organochloride pesticide and spray onto a farm, you will kill the bees, you will kill the ant, you will kill many other insects. But this very specific protein that is produced in the plant targets the very specific pests, the Lepidoptera and larvae. So when really a statement is made that they are grown using chemicals and more chemicals, that is not true.
Okay, what about what Anne mentioned, cross-pollination, right?
The potential of...
if I have a farm here, or small farmer here, who's using GMOs, and right next is organic, isn't there a chance that there will be some cross-pollination?
That's right. And that is one of the aspects that we look at when we are assessing environmental safety. We look at the potential of, it is called gene flow, the flow of genes from the GM crop to wild or even cultivated relatives. When we roll out the release of the GM maize, for example, we will follow it up with monitoring and technology stewardship to ensure that the two are able to coexist in the cropping system. Certainly, as Anne has put it, and also Mheshimiwa, the cropping system, our cropping system where you plant beans and pumpkins and maize and stuff on almost the same plot may present a challenge. And that is where now we would want to roll out this technology with the support of extension service, support of technology stewardship, and that is a commitment that has been given by the current government that we want to revitalize the extension system in partnership with the county government.
But Mheshimiwa, this matter could go to court, couldn't it?
Yep.
It might end up in court.
Might end up in court. And I just want my two colleagues to the right to understand that we are not raising these concerns because we want to be alarmist. We are raising concerns because many Kenyans are worried. Now, what the doctor in Zoom has asked, and that is the answer that we should be giving him, is that which trials have we done? Clinical trials for purposes of obviating any issue of harm to human nature, given the fact that we already saying we are unbanning GMOs, we are getting others from outside. I'm happy we've been doing research internally. We want to understand and get the landscape of maize, GMO maize, non-GMO maize, to see what we are talking about. What they are not telling us is that you will need the same challenge, you will face the same challenges with GMO crop as we face challenges with our normal crop. Whether they are not going to plant in Garissa just because there is not enough water. They are not just going to allow the field to be grown with weeds. And our major problem has been to manage the weeds. And that's why they were saying GMO is coming to solve that problem, because we can use some herbicides which GMO is going to be resistant to. But now they are not telling us Wanjiku has been weeding her farm twice a year. What are you telling her? Is GMO going to solve that problem of weeding? Or is she going to be exposed to the same harmful GMO elements without mitigation? The second issue, I want especially the person of biosafety to tell us what have they prepared in terms of the farming community, so that we don't take people by surprise. These are challenges that are real.
Yeah. Prof, let's deal with the clinical trials. Any done yet?
There are very many ways we deal with GMO production. And if she was talking about 8 years, to generate a GM product takes between 12 to 15 years.
To test it on humans?
I'm just giving the... And therefore, when they keep thinking that, that it's faster, it is not. It takes 12 to 15 years to generate a GM. And before you release that from the lab, which is contained environment, we check it and get to know it's fine for that specific trait. And probably, Jeff, let me demystify this GMO. I think in our mind, we are thinking GMO is this thing that will solve every challenge that we have. That is not true. If we want to target a GMO for drought tolerance, then what do? Scientists don't come up with DNA that doesn't exist. We only pick what already naturally exists. So there's nothing really new that we are creating in the lab. For instance, if you go to Northeastern, if our problem right now, which we know is drought in Northeastern, and we want animal feed, we first must look at where is the plant. Is there any plant currently existing in Northeastern that is tolerating the drought? Then that is where it all starts. And which crop or animal feed do we want to improve so that it survives there? You see how the thinking starts. Then let's say, for example, it is sisal. You know, it's a little drought tolerant. Then we say, our cows may not probably be eating sisal. So, but we need the gene that makes it drought tolerant. It could be the succulence of the leaves, or the roots are deeper so it gets a bit of water. You get? Those are only two sets of genes. So we don't need the bitterness, we don't need the thorn in the sisal. But when you take, say, maize to cross with sisal, it can't happen naturally. So what this gene, the technology does, is to say, this time round, we are dealing with the drought. And if we feel that we also want to deal with the pest at the same time, then we go for the BT, the Bacillus thuringiensis, which is also, interestingly, a soil-borne bacteria that we eat in salads. So when you hear people saying, how it will kill insects, will it kill us? First of all, the alkaline, the gut of the insect is alkaline, and that's what actually promotes it to stick, and then it swells, and it can't feed. So what we do, And that's why I agree with them, that when you pick a GMO product, you must ask, what is improved in this seed? As it is right now, the government has addressed, say, Jeff, if you want to deal with food security, you must look at several things. You must look at the soil, you must look at the seed, then you must look at the market, and then probably the systems for distribution and everything else. If fertilizers are now added, that they are treating the soil. Now you need to treat the seed. So what do you treat in seed? So you choose. Is it the pest that is a problem? Is it the drought that is a problem? Is it the disease like cassava? When you cut it, you find it brown in there. Is it the weeds like striga? They know them. Then we decide, what do we do first? Now we have the technology of picking tolerance for striga. We can cut it and stack them. You stack the drought tolerant, you stack the disease, like the disease tolerance, you stack the pest tolerance, and you can actually transform it once at a go. Then you will be able to have 3 different traits protected. And where are we getting the genes from? Probably that is what we need to ask. The genes are from already existing things that some of them we eat. So it doesn't become bad just because you have now moved it in maize. And I gave you an example. If you transform, genes from beans, because you want the proteins in beans to move into maize, naturally we eat them. Only that now, at the point when we are transferring these genes, there must be a problem with beans that beans can no longer grow. And we say we still need the proteins.
Okay,
That is the thinking.
Sure. Dr. Bernard Moya, I'm sure you're listening to this conversation. What are your thoughts right now? I mean, there's still so many unknowns here.
Jeff, yes, we look. Can I ask a question to a lady and gentleman together with yourself? What is the age of humanity? A lot of billion years ago, and I think the reason I'm posing that question is there is room for biotechnology to improve food safety and increase nutrients. But we seem to be rushing towards a biotechnology which is still young, which can, if it is realized, can cause perhaps serious issues to the health. And I'm just saying something small, simple. Look at what parts of the world they have done as far as GMOs are concerned. Are we in Kenya in a position to say, yes, globally, we have done this, we can see the results, and then the results are good, the impact is marked to humanity, and we use it? Why are we rushing? Biotechnology has got a very strong room in the future to do so much as food issues, food security is concerned. But when you look at issues in our world, particularly in where we are in Kenya, look at the issues. Somebody had just talked about something smart there, about the socio-economic issues related to food in Kenya for that matter. I think I've done studies I've been in Israel, and I can attest to the fact that it is a desert, but we have seen how Israel produces food to actually feed not only its citizens, but also feed others, even here in Kenya. So my question here is, why we have biotechnology which is a smart scientific endeavor. But the question is, why can't we subject it to various scientific research so that when we come out with something, a product, which will impact positively to humanity instead of rushing? Jeff.
Good question. Dr. Roy, let me ask you that. You can answer that question. And also, what Mheshimiwa had asked, have you sensitized the Kenyan farmer? And what's the rush? What's the rush?
Thank you, Jeff. In fact, when the applicants, for us, like I said earlier, we regulate. When the applicant is developing the crop, and I want to touch a little on what Mheshimiwa said, when you look at the 2 crops, there is no significant difference. We carry out what is called confined field trials, or rather the applicant is asked by ourselves to try it in confinement, so that we check to see whether it is doing that which they said it will do. We did that for maize at Kiboko. We did that for cotton at Moya, in Moya, and several other places. There are several locations. Now, after that, when we were carrying out that, because I want to touch on the sensitization, farmers are made to come and see. In fact, in Moya, when the process was going on during the confined field trials, these are very, very controlled with a fence and some trying to make sure that there is no intruders who come into the trials. People kept looking through and saying, oh, what goes on in there? And curiosity built until they were almost coming in to see what it is. So we invited them, or rather the Kari then, Karo, invited them to come on a field day to see the BT cotton vis-a-vis the non-BT cotton. We had to seek for security to ensure that they don't pocket the seeds and run away with them. They said, we have seen this and this, it is good. We want it now. Then we kept telling them, oh, no, this is trials and you can't have it. They wanted to have it. When we did the Kiboko trials, the CFT in Kiboko, the scientists, when they make the photographs, they put the cobs from the BT maize and also the cobs from the non-BT, and then make the picture. Farmers, when they saw the size of this and the size of this, in fact, some almost wanted to steal it from there. So the farmers out there are looking for solutions. And that trial is one of the ways of sensitization of the farmers. After the CFT, which is enclosed, we conduct what is called national performance trials. The varieties that have been genetically modified are grown in various corners of the country where the particular crop grows, of course, in controlled areas within Karo establishment, and then the observations are made whether they are actually doing that which is claimed that they will do. After that, appropriate varieties are identified and they are now released to farmers. We have done that for cotton. We have completed the process for BT maize that is protected against the maize stalk borer. In fact, everything is done. We were at the point where we were doing documentation to go to cabinet because of the ban. Now, with the lifting of the ban, BT maize is the one that can become available anytime once KEFIS registers the varieties that have been chosen, which will be owned by Karo and produced by any company, seed company that Karo will enter agreement with. So these are products by ourselves. We have checked the safety, the trials that Prof has talked about, and which I agree again with our colleague who is joining us online, Dr. Muya, that safety assessments, we do it on a case-by-case basis. You know, like I said earlier, Jeff, it is difficult to say all these are GMOs. If it is BT maize, we take it through on a we check the profile. One of the things that we check, we look at the chemical composition of the GM maize and the chemical composition of the non-GM maize and compare. We come up with what is called substantial equivalent. And we find that they are substantially equivalent, except for the protein that is being expressed by this, which has been modified.
Okay, the question is, why does GMO have such a bad name then? Why? I want to ask you that. In general, let's take a quick break and talk about that. You know,
why?
Why?
Yeah, that would be.
Is it pressure from multinationals, maybe? Is there pressure?
And our colleagues should not be defensive. Let us demystify this.
Let's demystify. I think I agree.
Yes. But let's first take a break.
All right.
What are your thoughts? Can we demystify GMOs? Keep tweeting at Koinange Jeff, at Citizen TV Kenya, the hashtag JKLive. JKLive takes a break. We'll be back in a moment.